All right, good morning and good evening, and everyone. Welcome to this month membrane trafficking webinar hosted by Society of Chinese Bioscientists and American. I'm pleased to introduce my colleague Long Li, an investigator of Peking University, who will host today's seminar speaker.
Okay. All right. It's my great honor to introduce Dr. William Prince. Is a long-standing figure in the field of lipid trafficking and organelle biogenesis. For the past 20 years, he has made some major contributions to understanding how lipid droplets traffic membrane contact sites between organelles and organelle biogenesis. Is currently the chair of the cell biology department at the UT Southwestern. Before that, he was a senior investigator at NIH. He earned his PhD with John Beckwith at Harvard Medical School, where he worked on protein folding and translocation. And then his postdoc was with Tom Rapport at Harvard as well, and where he shifted his focus to ER morphology and made some groundbreaking contributions to understand how the ER is shaped. Then he went to NIH to have his own lab focusing on lipid trafficking between organelles and the membrane homeostasis. In 2022, he moved to UT Southwestern and took the chair of the cell biology department. In recognition of his outstanding work, he has been selected as the keynote speaker at many prestigious conferences. He also served on the editorial boards of journals like Journals of Cell Biology, Developmental Cell, JBC. And today, Will is going to share his latest work on tube-like lipid transport proteins. So let's welcome Will.
Well, thank you so much for that kind introduction, and thank you for the invitation to speak here. I'm looking forward to this discussion today. So let me start sharing my screen here, and we can start. So yeah, today I'm going to talk about a subject that my lab has been working on for many, many years, about 20 years now, and this is the issue of how lipids are exchanged between membranes at membrane contact sites. And I realize that probably for this audience, it sort of goes without saying, but I think it's worth emphasizing at the beginning here how much our view of the intracellular communication has really changed dramatically in the last 20 years. And this has come about because we were thinking about how what we didn't understand 20 years ago was the role of intracellular contact, of interorganelle contacts, membrane contact sites, and all the myriad roles they play in cell biology. And so this is just sort of on the left here shows a textbook view of a cell and how its cells are often depicted with the organelles all separated from one another. But of course, we all know what cells really look like is like this. And so they're really jam-packed with membranes, and everything is in close opposition. And about 20 years ago, when I started working on this, really everybody kind of ignored this. And really the only people that were really thinking about contacts between organelles were people that were thinking about intracellular calcium signaling, because it was clear even then that close contacts between organelles were critical for this. But most of the rest of us just kind of ignored it. But in the last 20 years, there's really been a tremendous sea change. And now we understand that all organelles are in contact with each other all the time. And the organelle that I've spent the most of my career working on is the endoplasmic reticulum, which is one of the largest organelles in most cell types and touches everything. And so the list of things that happen at contact sites just keeps growing. And so these are very important zones for signaling, lipid signaling, calcium signaling, many different types. And now the number of membrane dynamics and other events that happen at contact sites just keeps growing. There's a number of things, including autophagosome formation, vesicular trafficking, vesicle organelle fission and fusion. These things are all regulated by contact sites. And so this list just keeps growing. And so it's been amazing to be part of this this field in the last 20 years when it's gone from sort of a little sideshow to really one of the center topics of cell biology. But my lab is largely focused on is still the issue of lipid transport and metabolic channeling that happens at contact sites. And just to tell you a little bit about what are the sorts of things that cells use this trafficking for. So we've sort of divided lipid transport between organelles into two broad categories. So one is bulk transport. So this is the transport that's necessary to generate whole organelles. And a great example of this is autophagism formation, where when autophagism formation, mitophagy is induced in cells, these structures can be generated in minutes and require the transport of millions of lipid molecules in just a short amount of time. And so this transport is thought to require lipid transport proteins that I'll talk about in a minute. These proteins also are very important. It's becoming clear they're very important for membrane repair. But there's also sort of lower scale lipid transport that happens that's more about sort of tweaking membrane lipid composition or maintaining a particular composition of lipids in membrane. So some of this has to do with lipid signaling. So PIP signaling requires the trafficking of lipids between the ER and different organelles, particularly the plasma membrane, the Golgi. Non-vesicular lipid transport can also be used to enrich particular lipids in an organelle or de-enrich them. And it can also be used by cells to drive membrane formation, membrane domain formation. So how does this transport happen? So a typical eukaryotic cell expresses 20 or 30 lipid transport proteins, and there are many families of these transport proteins. And largely, they're not structurally similar to each other, but they all are conceptually similar in that they have a pocket or a groove that can usually bind just one lipid at a time, often covered by a lid or a domain that shields the bound protein from the aqueous solution. And this allows these proteins to shuttle lipids between membranes. So they can pick up a lipid from one membrane, shuttle it through the aqueous phase, and deliver it to another. But one of the big sort of changes in the field that's happened in only the last 5 years or so is that it's become clear that there's lipid transporters that seem to work by another mechanism, and that is that they form lipid channeling bridges or conduits that allow, or at least are thought to allow, lipids to flow between organelles. So before talking about those proteins, I just want to say sort of a basic fact that I think most people know, but a lot of people don't realize, which is that what these transporters are doing, none of them require energy, and all of them, you can think of them in a certain sense of working like catalysts. And what I mean by that is that they're taking a reaction that happens spontaneously. All lipids will move between membranes spontaneously. These rates are usually incredibly slow, too slow to be physiologically relevant. And it's been known for many years that the rate limiting step of this spontaneous movement is desorption, the rate at which a lipid will leave and move into the aqueous phase. But sort of pioneering studies from nearly 30 years ago showed that the rate at which this happens is basically determined largely by the hydrophobicity of the lipid. So the more hydrophobic a lipid, the slower this process happens. And it's important to remember that these lipid transfer proteins are just taking a process that happens normally and speeding it up. So this new family of proteins, which has a number of different names, and I'll call the VPS13-like proteins, are all very large. These are structural predictions. The structure of none of these proteins has been completely solved. These are all structures. These are structures predictions that we did with AlphaFold. They're all very large proteins. They're about 3,000 to 5,000 amino acids, and they have large twisting beta sheets that run the length of the protein. So these are very long. These are big enough to completely span a membrane contact site, which are usually on the order of, depends on a condition, but roughly 30 nanometers across. And it's easier to get a little bit of a sense of this tube when you look at this is a movie, and you can see that it's actually not a tube. It's more like Karen Ranch has described it as a taco, but it's like a slot that a lipid, at least in theory, is thought to be able to move along from one membrane to another. So one membrane would be here and the other here, and this would let lipids move from one to the other. So these proteins, so there are 5 in yeast. I just showed you those. In humans, there are 10, and they're all localized at contact sites, not surprisingly. And this is a nice review, a cartoon from Pietro Di Camilli's review, which shows the location of many of these different proteins. So the two that another interesting thing about these proteins is that disease mutations in many of these proteins, most of these proteins cause rare diseases, or in the case of this protein here that I'll be talking about today, seem to have an important role in a number of cancers, especially breast cancer. So I'm going to be talking about these two members of this family today, mostly. And they have this one, BLTP1, has this other rare neurodegenerative disorder that's caused by it. So, but one of the things, before I talk about those proteins in details, one of the things that an issue that my lab has been interested in for a very long time is how mitochondria obtain the lipids that they need for membrane biogenesis. Because mitochondria, there's some vesicular trafficking between mitochondria and the rest of the cell, but it's not thought to be a major factor. And that the idea is that most lipids, has been thought for many, many years, that most lipids that are needed to create a mitochondria have to come from the ER or other organelles. And in yeast, it's thought that this requires the Hermes complex, which is a complex I'll talk about in a second, a tethering complex that also is thought to transport lipids. And then also VPS13, which is one of these tube-like forming proteins, also has contacts between the vacuole, which is the yeast equivalent of the lysosome, and mitochondria. And when these proteins were first discovered in the last few years, I thought, well, these must be the proteins that are really responsible for moving high volume movement of lipids to mitochondria. And I should say that beautiful work from the Kukulski lab showed a few years ago, this is a paper in Nature, that the different components of the Hermes complex probably, even though they're not related to these at all, probably also form a tube that allows lipids to move from one organelle to another. And we also noted, and I'm not going to show you the data, but that 3 of these 5 proteins in yeast are also at contacts between ER and mitochondria. And so we thought that perhaps these are the proteins that mediate the bulk transport here. And so to test that, what we want to do is make a mutant. This is the beauty of working in yeast, make a mutant that's missing all of these proteins. And so we did that. We made a mutant that interestingly, you can make a mutant that's missing all 5 of the VPS13 tube-like proteins, and the cells are still viable. And then you can also use the degron to get rid of one of the components of the Hermes complex. And it's been known for a long time that if you get one of the components, the whole complex falls apart. And so we can make a cell then that when we degrade MDM12, which is one of the components of the Hermes complex, and it's also missing these 5 parts of these components of these 5 lipid transport tube-like forming proteins, so it doesn't have any of the tube-like proteins that are in yeast. And then we can estimate bulk lipid transport to mitochondria by looking at the rate of synthesis of cardiolipin. So cardiolipin is a lipid that's only synthesized in mitochondria, it's synthesized on the inner membrane, and it's been known for some time that the precursors for cardiolipin that's synthesized in mitochondria are phosphatidic acid and CDP-DAG, so 2 lipids that are synthesized in the ER. And so if there was a complete block in cardiolipin synthesis, then that would indicate there was a complete block in bulk lipid transport to mitochondria. But remarkably, that doesn't happen. There is a decrease, but it's only about a 40% decrease. So this is really a shocking result to us. And I think what it suggests is that I think these proteins, of course, do transport lipids to mitochondria, but that there must be other proteins that do it as well. So this was really surprising. And I think one thing we could talk about later is, what does this mean? Because it does suggest, I don't think there are any more proteins in yeast that are these tube-like proteins, although it's possible there's others that we haven't detected somehow. But then it suggests there must be other mechanisms, very efficient mechanisms for moving lipids from the ER to mitochondria. So the question is, if they don't do this bulk transport to mitochondria, or they only do that somewhat, what do these proteins do? And so in a paper that we've already published, and I won't talk about too much, we think that one of these 2 proteins, which is one, it's called CSF1 in yeast or BLTP1 in humans. BLTP stands for bridge-like transport protein. What we think this protein does is we think that it moves PE to the ER to support GPI anchor biogenesis. So GPI anchors are lipidic anchors. Proteins are attached to these lipidic anchors in the ER. This is conserved from yeast to humans. It's a very long biosynthetic pathway. I'm just showing part of it, but each GPI anchor has 3 ethanolamine phosphates on it. And interestingly, it was shown many years ago by Anant Menon's lab that these ethanolamine phosphates aren't, so this is a process that's all happening in the lumen of the ER, and these aren't free, they come from the head groups of PE. So PE has an ethanolamine phosphate group. This is the phospholipid, phosphatidylethanolamine, and there's 3 enzymes that move this head group onto the forming GPI anchor. And so, so that means that each GPI anchor requires a PE molecule. And again, without showing so much of the detail, we did this is work we did mostly in yeast, where we can show that cells that are lacking this protein, which is called CSF1 in yeast, this bridge-like protein, build up this intermediate, lipid intermediate, which has the name 31A. And so 31A has the anchor, but it's missing the ethanolamine phosphates. And so what we think is that this is also true in humans as well, we think. So there's the human homolog of CSF1 has these various names, and also BLTP1. And so in human cells, you can look to see if there's a defect in GPI anchor biogenesis, because you can by looking to seeing whether there's a decrease in GPI anchored cell proteins on the cell surface, which can be done by FACS sorting. And so what we can see here is that here's a control where we knock down one of the proteins that's involved in GPI anchor biogenesis, and so we see a decrease in here. This is a protein called CD55, which has a GPI anchor, and we see a decrease also when we knock down this gene here. So we think that this is conserved. This function of this protein of supporting GPI anchor biogenesis is conserved from yeast to humans. And so the model then of how we imagine CSF1 to work, or in humans it's BLTP1, is that it's moving PE from another organelle. Either this protein is mostly localized at contacts between the ER and plasma membrane, and we think that it's moving PE from the plasma membrane and perhaps other organelles. And we're also able to show in this paper that it directly interacts with this protein called MCD4. So this is one of the enzymes that transfers ethanolamine phosphate from PE onto the GPI anchor. And so we think that the protein may directly channel PE to this enzyme. And so, but one of the really interesting things about this was that this seems to only be important. CSF1 is only important for GPI anchor biogenesis in conditions when the Kennedy pathway is not operational. So in yeast and in humans, there are 2 main pathways by which cells synthesize PE and PC. So these are the 2 most abundant lipids in most membranes. One is called the Kennedy pathway, where ethanolamine or choline are used directly to synthesize these lipids. And the other one in yeast is called the CDP-DHE pathway, where the PE is synthesized from PS. And so in yeast, it's possible to have a condition where this pathway is essentially off or on. And so when we add ethanolamine, then this pathway is on. And interestingly, we see that this precursor doesn't build up at all when the pathway is on. So what that suggests is an interesting, a surprising result, which is that this pathway seems to be important, particularly under conditions when the Kennedy pathway is not operational. And so we think that it's particularly important for bringing PE back to the ER under conditions where cells can't make PE by this pathway. And this is actually quite surprising, and we're still trying to understand it, because there's PE in the ER all the time, and so it isn't 100% clear why this pathway is particularly, when this pathway is operational, cells need to move PE back to the ER to synthesize GPI anchors. But the story that I want to talk to you about most today is about this other protein. So this is still unpublished work. This is another member of this family. It's called BLTP2. And it turns out that what we think it does in cells is remarkably similar to what BLTP1 does. So just to show you, so BLTP2 has two homologs in yeast. The homologs are called FMP27 and HOB1. This is just showing an overlap of the predicted structures of FMP27 and BLTP2 to show you that at least the prediction structures are quite similar, and these two proteins are good homologs. And so when we first started working on this protein, we didn't know what it did, and it was actually much easier to find phenotypes and work on it in yeast. And as I'll show you later on in the talk, all the things we found that it does in yeast turn out to be conserved in the human protein as well. And so the first clue that my postdoc, Subra Banerjee, got about how this protein might work is that he found that it doesn't grow at very low temperatures. So if you're not used to looking at these are dot blots where these are serial dilutions of yeast, and this is just a way to look at how well the cells grow. So normally yeast grows at 30 degrees. Here we're growing at a very low temperature, 18 degrees C, but yeast can still grow. But cells that are lacking this protein, FMP27, have a severe defect in growth. And one of the interesting things that we found, which is related to the story I just told you before, is that growth can be restored when we add ethanolamine to cells. So remember, so this is a media that normally lacks ethanolamine, and so the Kennedy pathway is off. But when we add ethanolamine to these cells, now they grow just fine at low temperature. But interestingly, they still don't grow even when we add choline. So this suggests that it's really a defect in PE production that's important for that these cells have some kind of defect in PE production, or as I'll show you in a minute, what we think is it's a defect in the distribution of PE in cells. So we wanted to understand PE is also a precursor for other things, particularly PC, but it's also used in other reactions as well. And we wanted to understand if what we are seeing here, was it really a PE story or was it a derivative of PE? And so one thing that we were able to make use of is that for both yeast and for higher eukaryotes, they can make a lipid that's very similar to PE, but instead of having ethanolamine, it has this propanolamine group. So this is just like ethanolamine, but it has one more carbon. And the remarkable thing about this is that cells will use it and make a lipid that has using this as the head group, but then this is metabolically inert. Cells can't do anything else with it. So they can only make this. And what we were able to show is that when we add propanolamine to the media, this also lets them grow just fine. So this says that this is really a PE story. And I should also say that PE is a lipid that has biophysical properties that are different than most of the other most abundant cholesterol phospholipids in cells. It's a non-bilayer forming lipid. And so that's also true of the lipid that's made from this. So we think that this has to do with the biophysical properties of PE that are particularly important for complementing this growth phenotype. So we were puzzled about what this could be, but what we think this is about is we think it's about membrane fluidity. So as many of you probably know, one thing that cells do when to adjust to changes in temperature is they have to maintain the fluidity of their membranes. And one way they do that, the most commonly known way they do this, is by changing fatty acid saturation. So the degree of saturation determines the fluidity of the membrane. But another way that cells do this, and people have realized this in the last few years, is they can also affect membrane fluidity by changing the ratio of PE to PC. Or really, it's another way to think of this is the ratio of PE to all the other lipids in the membrane, because PE is a non-bilayer forming lipid, and so it's thought that the ratio of these two lipids to one another affects fluidity. In fact, it's been shown. So we wondered if that's what this was about here, and so so first we looked at the ratio of PE to PC in cells that are missing the wild type cells or cells that are lacking FMP27. And so what we found is that at 30 degrees, when you move a wild type cell from 30 to 18, the ratio of PE to PC goes up, but that fails to happen in cells that are lacking FMP27. But when we give them ethanolamine, so that is they can make PE by the other pathway, then this defect goes away. So this is a defect that only happens when the Kennedy pathway isn't operational. So we also, by fractionation, we could show that this is a defect that particularly affects the plasma membrane. So if we look at this is now looking at 18 degrees C. Well, if we look at the ratio of PE to PC, in the endoplasmic reticulum, there's no difference between wild type and FMP27, but there's a huge difference here between wild type and cells that are lacking FMP27. So this is a defect what's most affected here is the ratio of PE to PC in the plasma membrane. And so this suggests the model then to us. So I should say that FMP27 has been localized by us previously at contact sites between ER and the plasma membrane. And so the idea here is that at low temperature, FMP27 may facilitate the movement of the PE from the ER to the plasma membrane to maintain plasma membrane fluidity. So we can look directly at the fluidity of the plasma membrane by using probes like this. So these are probes that so really I should say we're actually looking at the fluidity of the outer leaflet of the plasma membrane, not both leaflets. These are probes that just enter the outer leaflet and aren't flipped into the rest of the cell. And so by looking at, we can use a fluorescence anisotropy to look at membrane order using these dyes. So at 30 degrees C, there's no difference in the fluidity of the plasma membrane between wild type cells and cells that are lacking FMP27. But when we look at 18 degrees C, we see that the membranes are more disordered in cells that are lacking FMP27. And again, this defect is corrected by adding ethanolamine. So this is consistent with the model that FMP27 then is controlling the fluidity of plasma membrane at low temperature, specifically by bringing PE from the ER, where it's synthesized, to the plasma membrane. So another thing we did is we did lipidomics to look at whether FMP27 could also be regulating fluidity by affecting acyl chain saturation. And so this is lots of data, but the short version of this data is that the answer is no. So this is maybe the easiest way to make sense of all this data is just we can do an unsaturation index, looking at the total unsaturation of all lipids in cells and so this is when you look at a wild type cell, the saturation index goes up at 18 degrees. So that's a well-known mechanism that cells use to maintain fluidity. And that's true. There's no defect in that in cells that are lacking FMP27. So what this suggests is that what FMP27 is doing, the way that it's affecting plasma membrane fluidity, is specifically by moving PE and not by affecting acyl chain composition. So the next question we ask is this also true of the human homolog of FMP27, which is called BLTP2? And what I'm about to show you is that remarkably, all of this is conserved. So this is actually really surprising to us, and at least initially, because of course, human cells don't grow at 18 degrees C and they don't need to adapt to temperature in the same way. But I'll tell you what I think it does later on. So the first question we wanted to ask is BLTP2 actually at these junctions? So there's been some hints before, but we wanted to demonstrate that BLTP2 is actually at ER plasma membrane junctions, just like the yeast homolog FMP27. So there had been a previous paper from the Barashula group, which worked on the BLTP2 homolog in flies, which has the name Hobbit, or they call it Hob here. And this is a marker of contact sites between the ER and plasma membrane. They're looking in flies, and they can show here that Hobbit goes to these sites. We did a similar kind of experiment. In this case, we looked in U2OS cells. We're using a marker of ER plasma membrane junctions that is called MAPPER. This was made by Jen Liu here at UT Southwestern. And what we can see is that BLTP2 goes to these sites, these junctions of ER plasma membrane junctions. So that's similar. That shows that BLTP2 has the same localization as the yeast homolog. And remarkably, we found the same sort of effects of ethanolamine and propanolamine on cells. We made a HeLa strain that had a knockout of BLTP2. These cells have a growth defect. They grow poorly. I'll talk about this more later. So they grow poorly in media when we grow them with dialyzed FBS. So that's FBS that lacks ethanolamine. They grow poorly, but that growth can be restored when we add ethanolamine. So this is just like yeast, and it also is restored. In fact, it's restored even better when we add propanolamine. So remember, this is the precursor that allows cells to make a PE-like lipid, but a lipid that's metabolically inert. So this says that just like the yeast mutants that are lacking FMP27, cells that are lacking BLTP2 have a growth defect that can be complemented by making more PE or a PE-like lipid. So we think that we know that BLTP2 also regulates plasma membrane fluidity in the same way. So we can look at total PE in cells, and it goes down in cell. This is, again, I'm only showing the data for in growth of the cells in media that lacks ethanolamine. None of this is correct. This is the same when cells are grown in media with ethanolamine, but without ethanolamine, they have a low level of PE. The ratio of PE in the plasma membrane, just as in yeast, also decreases in cells that when we knock down BLTP2. And similarly, just as in yeast, we can show that there's no change in the distribution of acyl chain composition. So this is a very complicated graph, but the short version here, we're just looking at wild type, the BLTP knockouts, or with or without ethanolamine. And just the short version of this, again, is that there's really no significant difference between any of these cases. And so, again, we think that BLTP2, like its yeast homolog, isn't regulating or affecting lipid acyl chain composition, but what we think it's doing is it's modulating lipid membrane fluidity by affecting the transport of PE. And so, again, using the same, this is a different dye, but it's the same idea. We can do the same kind of experiment that we did in yeast to look at the fluidity of or the rigidity of the plasma membrane, and we see that when the knockouts are grown in conditions that lack ethanolamine, we have a significant increase in fluidity, and that's corrected when we add ethanolamine. So again, this is remarkably all the same. All these properties of what we think BLTP2 is doing are just the same as the yeast homolog. And I also want to just say, as a side note, I think this still, I still think that yeast is a very useful model or tool, and we were able to find all this out in yeast much more quickly than we could have done in mammalian cells. And it's a great tool because then we're able to show that what we found in yeast is conserved in humans. So as a further hint that we're on the right track here, that BLTP2 really is involved in controlling membrane fluidity, we noticed we used DEPMAP. So DEPMAP is a big a collection of many all genes have been knocked out in a number of different cell types, and then you can look for codependencies. That is, when you knock out two genes, knock down two genes at the same time, which ones are additive. And when you look at this list for BLTP2, the number 2 hit here is this protein called TLCD1, and TLCD1 is a protein that is also plays a role in a pathway that modulates membrane fluidity, particularly plasma membrane fluidity. So TLCD1, together with its partner called TLCD2, regulates fluidity. These are proteins that are actually in the ER. They're thought to modulate MUFAs. So these are multiply unsaturated fatty acids, particularly into PE, and they might operate at contact sites. How these proteins modulate membrane fluidity is really not well understood. But what we can show is that when we have a knockdown, when we start with cells that are lacking TLCD1 and then knock down BLTP2, they grow very, very poorly. So in a sense, confirming this result here. And we can show that when we start with these cells and then knock down, so cells that are lacking TLCD1 and have a knockdown of BLTP2, we have an additive effect on the membrane fluidity. So we think that this is more evidence that BLTP2 really is involved in regulating membrane fluidity, and we think that these are two different pathways. So we don't know how TLCD1 works, but we hypothesize that the way that BLTP2 works is by modulating the fluidity of the plasma membrane, specifically by transporting PE from the ER to the plasma membrane. So this in yeast, of course, we think that this is important for cells adapting to growing at very low temperature. In humans, of course, that's not really relevant. And so we wondered, under what circumstances is BLTP2 particularly important for growth? And we noted, again, using this resource DEPMAP, that we can, this is a wonderful resource where you can look at many different cell types, and that when you look at at cells that are, so you can look at cells, cancer cell lines where BLTP2 is overexpressed. So this is expression index of BLTP2, and then how well the cells grow. And so for many of the breast cancer cell lines, particularly invasive breast cancer cell lines, these are the 3 that are near the top here. So they grow very, very poorly, and BLTP2 is expressed. So it seems to be near essential for growth of these metastatic breast cancer cell lines. We used this triple negative breast cancer cell line to show that when we knock down BLTP2 in these cells, it makes them grow very, very poorly. So this is a control epithelial cell line. So it's BLTP2 is not required for the growth of these cells. And this is just 2 different assays that are looking at growth. And I should say that we see these defects only and specifically when we use media, a growth media that lacks ethanolamine. So again, this is the same story again, that this only seems to be important under conditions in which ethanolamine is not present, or there's very low levels of ethanolamine. And another interesting thing about this protein is that it's pro-survival for the triple negative breast cancer cell line. That is, when we knock it down, we induce apoptosis. We're just looking at early and late apoptosis. So this protein is important for cells for the growth of cells, and we think it's particularly, it induces, when it's lacking, you get the spontaneous induction of apoptosis. So we wondered whether the BLTP2 is particularly important for the ability of these cells to grow invasively in organisms. And so we used a xenograph model. We used a zebrafish xenograph model. We did this in collaboration with the Fiocco lab here at UT Southwestern. And in this model, we use young zebrafish to look at the ability of cells to metastasize and grow in an organism. In this case, the cells are injected here near the yolk sac, and then you can look at them go through the rest of the organism, travel through the rest of the organism, and metastasize. So this is a very nice system because it's relatively easy to do, and the Fiocco lab is known for its microscopy, and they do this on a lattice with a light sheet microscope. And what that allows them to do is we can track every single cell and how it moves through the organism. So it's really quite a remarkable resource here. And so what we can do is we can make maps that look like this. So this is showing many, many different cells added up. The greater number of cells is, if you take the animals and overlay many of them on top of each other, you can ask where the cells go after injection here. And so when we look, this is 5 hours post-infection, 24 and 48. So this is control. This is the control when we inject the triple negative breast cancer cells here. They spread throughout the animal. Here we're looking at, you can see them growing in the tail and metastasizing. And there's a huge defect here in the cells where we have knocked down BLTP2. And this is a quantification of the number of metastasis in the tail. So if you look at the control, they're up here. This is over, of course, averaging many, many animals. And over time, the cells that are lacking, where we've depleted the cells of BLTP2, we have a big defect in metastasis. And sorry, there's this is another measure of looking at metastasis here at the top of the animal. And again, you see a huge difference between the control cells and the cells that are lacking BLTP2 over time. So what this says is that BLTP2 is necessary for, we think it's necessary for invasion. Here's I'm sorry about this. So here's stills from movies that we can take. We can watch the cells actually traveling through the fish. These are images of the same fish, just the fish here are expressing a vascular marker here, which is shown in blue. So this is showing the vasculature of the fish. And these are the triple negative breast cancer cells that are expressing F-tractin. So this is a probe for filamentous actin. And so when we're looking at the normal triple negative breast cancer cells, you can see them growing. These are the actin rings. This is what you see in cells that are actively invading other tissues. And when we look at the animals that have been infected, that xenografted with the triple-negative breast cancer cells where we've knocked down BLTP2, we see very few of these structures. So we think that this is, at least in the case of these cancer cells, what we think this is important for is that it's maintaining the proper function of the plasma membrane. So these invasiveness and forming these filamentous actin structures are very important for invasion. And we think that that's why, at least in these particular cancer cells, types of cancer cells, BLTP2 is important for maintaining proper function of the plasma membrane. So our model then is that BLTP2, or in yeast it's called FMP27, moves PE from the ER to the plasma membrane to help maintain plasma membrane fluidity or rigidity. So in yeast, we think this is a pathway that's necessary for cold adaptation, for maintaining fluidity when the cells are transferred to low temperature. In humans, this seems to be particularly important for the growth of some cancer cells. It also is pro-survival, and we suspect that it's particularly important for cell invasion. And so a big question then is that we think that in the case of in triple-negative breast cancer cells, and the question is, why are they particularly dependent on these? And we suspect, what we don't know, is that they may grow in environments that are particularly low in ethanolamine, and so that's why they require this pathway to maintain membrane fluidity under conditions where they're not making PE from the Kennedy pathway. So what's next? There are both mechanistic and important functional questions that still remain to be answered. So, I mean, one of the main questions here is that in both cases, for BLTP1 and BLTP2, we speculate. All the data suggests that these proteins are transporting PE. It certainly looks like it, but we actually have yet to demonstrate that they really do transport PE. And this is an incredibly difficult problem because these are gigantic proteins and they're hard to purify and work with. But we want to know, do they really transport PE, or maybe they transport something else and they're indirectly affecting PE? And another, of course, major question for these and for all this family of proteins is nobody's demonstrated conclusively yet for any of them that does PE or other lipids actually flow through the center of the protein. That's what they certainly look like the proteins are designed like that, but it isn't. Nobody's really shown that that's true. And of course, a major question here is how is this transport regulated? Because we think that if these are in a sense big passive tubes, we think that the most important part of these proteins will be their interacting partners, and which regulate either the movement of lipid into or out of these tubes. And then another major question for this and for many other lipid transport proteins is what's driving the transport? What drives PE from or any lipid from one membrane to another in this case? So there's lots of questions about the functions too for BLTP1, which is the protein I just talked about a little bit at the beginning, which we think is involved in GPI anchor biogenesis. How does this lead to neurodegeneration? The mutations in this cause this rare neurodegenerative disorder. And as I was talking about before, another question for BLTP2 is, why is it particularly important for some cancers? And we suspect that this is related to ethanolamine metabolism or availability. There's already lots of hints in the literature that breast cancer cell lines in particular have defects in ethanolamine metabolism, but how is that related to this, or is it the cause of making them particularly dependent on BLTP2? We don't know yet. And then what I'm saying down here, we wonder why do cells need these? And we think that they may be particularly important under conditions where PE in the ER is low. And the vast majority of PE that's made in the ER is converted to PC. And so it may be that these proteins are necessary under conditions where most of the PE in cells is used and converted to PC. And so that's why we suspect that in cells when we have defects in PE metabolism by the Kennedy pathway, PE levels in the ER are low. Most of it is used to make PC, and the ER may be in some sense starved for PE, although we still don't know whether that's the case. So I'd just like to end by thanking the people that did this work. This is a picture of my lab soon after I got here. It's actually somewhat bigger now. Here's our collaborators from both here at UC Southwestern and around the world, and and our funding. And I thank you for your attention. I look forward to your questions and discussion. Thank you.
All right, very interesting talk. So now time for questions. Should we start from the chat, please? Okay, I'll start from first question. There's are there any changes in the iso chain of the phospholipid in the FMP27 lacking cells? I think one of your slides. Answer that.
Yeah,
Right.
Yeah. Then it's from Ehong. Is cold temperature activate FMP27 or simply increase ERPM contact?
Yeah, I think this is a really important question. We do know that at low temperature, the expression of FMP27 goes up quite a lot. So it's 4 times more abundant at low temperature than it is at 30 degrees, which is normal temperature. The localization doesn't change. What we're really interested in understanding also is, do its interactions partners change under these conditions? Because we suspect that it does, which is, in a sense, what you're asking is it in a sense turned on during these conditions? And so we suspect it is, but we don't know yet. Yeah.
And Yihong, I think you have more questions. Probably you can ask directly.
Yeah, thanks. Okay, I'll take this opportunity. So yeah, I also wonder what happened if you return the cells from cold temperatures to normal temperature. So are those extra Ps being transported back to the ER, or they are somewhat gets extracted or degraded?
That's a great question. We know that the protein level goes down quickly, but we don't actually know what's happening to the PE level. That's a good question. We haven't addressed that. The prediction should be that it would need to get moved back to the ER or get degraded or turned into something else. But we don't know. That's
kind of related to this. Is, do you know or do you speculate any? So this protein, if it's actually mediating lipid transport, is it bidirectional or just unidirectional?
Again, questions we really wish we knew the answer to, but I mean, the model all fits it being unidirectional, right? But I mean, the way that the proteins look, there's no reason why they couldn't move lipids one way or the other, right? But I mean, this is a big question, not just for these proteins, but for everybody that's working on these fields, on these proteins. The best study of these proteins is ATG2, because you can imagine lots of people care about this because it's so important for autophagy. But even then, it's not really clear what drives transport and how do you get directionality out of it. So this is a really important question. Yeah.
Maybe lastly, I want to just ask you things about this neurodegenerative disease causing mutations. Have you ever tried to express these mutants in your loss of function cells to see if it still can complement the defect?
You mean the disease-causing mutations? Is that what you're asking?
Yeah,
that's right. They don't complement, right?
They don't complement.
That suggests that we're on the right track.
Okay.
Here.
That's great. Right. All right, that's all my questions. I'll ask Long to take over.
So may I ask?
Okay.
Okay,
go ahead. Yeah, go ahead.
So Will, so how you see how the fluidity is playing here? So if this protein has no fluidity, so how fluidity is playing here? Should we think about membrane as a kind of globally, or can changes in fluidity control domain organization of the membrane?
Yeah, I think this is a great question and something we're not really sure how to get at, right? I mean, of course, the plasma membrane should have many different domains, right? And so it might, all we can look at with these probes is just sort of global fluidity, and wherever they're going, we don't even really know which domains in the plasma membrane they're going to. But that's a great question. It may be that this is more about moving PE to particular domains as opposed to just bulk moving PE to the plasma membrane. We'd love to know the answer to that, but the problem is we don't have a good way to do that. It's hard to look, at least with the tools we have right now. It's very hard to look at those domains in the plasma membrane.
And another question is whether you see any. So you looked at tails, at tails. Do you see any changes in polar heads, like kind of compensating for the lack of these proteins? So are there any changes in polar heads?
In the polar heads? Well, I mean, largely no. I mean, the biggest changes are different. I mean, there is a decrease in the total amount of PE, right? And
yes,
for reasons that seems to be the major change, right? That's why we still think it's a PE story, even though we haven't shown that the proteins actually move or even bind PE. But the main change seems to be in the level of PE and the amount of PE that's the plasma membrane. The acyl chains don't really change, and the other the other polar groups are largely the same. There's not they're mostly
There
the
were old studies from Ben Cruyff lab in which they worked on E. coli, and it's very simple situation. There are just 3 lipids there in effect, and when they in effect knocked out phosphatidylethanolamine, they had more of cardiolipin and calcium dependence, presumably because cells were trying to grow, trying to compensate for the lack of one lipid of negative spontaneous curvature, phosphatidylethanolamine, by having other lipids which can compensate in terms of curvature. So that's what I mean in terms of other lipids changing.
Yeah, it doesn't seem like there's much of a change. I mean, it mostly seems to be a change in PE. That seems, in terms of lipids with negative spontaneous curvature, right? DAG is about the same.
Right.
PE is about the, so that's what we think. But it's still a good question. I think it's still possible there's other subtle changes that we haven't seen yet.
Right.
That's right. Yeah.
Yeah, we are a great talk. So I'm curious about the PE transport from mitochondria to ER. So does mitochondria make a lot of PE?
Well, there's a PE synthase in mitochondria. So it's called in yeast, it's called PSD. So this is phosphatidylserine decarboxylase, and this is conserved from yeast to humans, right? And so what we were able to show, and I didn't show the data, this is talking about the protein called BLTP1 or in yeast CSF1, is that it doesn't seem to be required for, so you can grow yeast in a condition where the only way they can make PE is in mitochondria. And that means that all the PE has to come back. But when you knock out CSF1, the cells are still okay. And so that says that there must be other mechanisms that are able to move PE. That's why we think that it's, for reasons we really don't understand, this really seems to be specific for GPI anchored biogenesis, right? And as opposed to just bulk PE transport. But yes, so we could talk about it later, but there's ways you can make yeast that the only way they can make PE is by this enzyme that's in mitochondria. So that means by definition, every PC molecule had to come from PE that moved that. And so, yeah, but so it can be really, it can really be a high volume transport. Yeah, that's right.
Yeah.
Okay, so there's another written question from Marini, and I also I also wonder what happens during stress heat shock response, chaperone involvement, which also play roles in cell functions in neurodegenerative diseases, intracellular transport traffic, proteostasis, organelle associations. Thank you.
I mean, this is a great question. I mean, there are many labs working on this, not us as much, but yes, I mean, of course, there's many changes, and what we want to understand is how these changes are integrated with lipid trafficking. Lipid trafficking is only a teeny part of what's happening here during neurodegeneration, and we want to understand the link between nonvesicular lipid trafficking and these other processes that are being studied by lots of labs. It's a good question, though. Yeah.
Okay. All right. And more questions?
Thank you. Thank you.
Thanks.
All right. Then, if no more questions, let's thank Will again. Great talk. Thank you.
Thank you. Thanks, everyone.
Thank you, Will.
I enjoyed it. Thank you.
Yes, thank you.
All right.
Nice seeing you again.
Okay. Yeah.
Bye-bye.
Okay. Take care. Bye-bye.
Yeah. Bye.